The overall goal of this procedure is to induce hepatocyte ablation in zebrafish for the analysis of biliary-driven liver regeneration. This is accomplished by first exposing transgenic larvae that express nitroreductase in their hepatocytes to metronidazole, or MTZ. In the second step, the level of hepatocyte ablation is assessed based on the relative size of the liver after MTZ treatment. In the final step, fully ablated larvae with a tiny liver are collected. Ultimately, the size of the liver and the expression of liver markers in the regenerating liver are measured by epifluorescence and confocal microscopy, respectively.
The main advantage of this technique over existing methods like rodent oval cell activation models is that with this technique, the regenerated hepatocytes are mainly derived from biliary epithelial cells.
This method can help answer key questions in liver regeneration field, such as how do biliary or liver progenitor cells contribute to the liver regeneration.
Generally, individuals new to this method will struggle because the liver size in ablated and regenerating larvae is variable.
To conduct timed matings, set up adult male and female hemizygous or homozygous fish overnight with a divider between them. Remove the divider the following morning when mating is desired, using a fine plastic mesh strainer to harvest the embryos. Turn the strainer upside down and use a squeeze bottle to rinse the mesh surface with a fine stream of egg water. Then transfer up to